Hello friends, welcome back to my YouTube channel Best Online Medical Lectures. In the last 3 videos, we have discussed about the very important hormone insulin. So we have discussed about the structure of the insulin, synthesis, its secretion, its release, that is the secretion, and also about the regulation of secretion of insulin hormone. Okay, today we are going to discuss about the mechanism of action of the insulin on its target cells, how insulin will act on its target cells. So as you know, insulin is secreted from the beta cell of the islets of Langerhans into the bloodstream, okay, from the beta cell of islets of Langerhans from the pancreas, okay. So once it is released into the bloodstream, it will go to the all our body cells, but the target tissues or the target organs of the insulin action is liver, striated muscles, that is the skeletal muscles or cardiac muscles, and the adipose tissue, okay. So this is the target cells or the target tissues on which mainly the insulin hormone will act, okay. So we are going to study today how exactly this insulin, when it is released into the blood, will act on its target cells on the liver, skeletal muscle and adipose tissue, okay. So for this I'll just draw an hypothetical cell on which insulin will act. So consider this as a cell, okay. A cell will have a cell wall, its cell organelles like nucleus and other structures, okay. So consider this as an hypothetical cell, okay. Now with the blood vessels, that is from the blood vessel on this target cell, this insulin molecule will act on this target cell, okay. So basically the action of the insulin is on the metabolism, that is the carbohydrate metabolism, protein metabolism and the fat metabolism. So basically the insulin will lower the blood glucose level on the by its action on the target cell. So how it will take do that action, we are going to discuss today. Now this insulin molecule is a larger molecule, larger protein molecule having a 51 amino acid protein molecule, okay. So and it is a very polar molecule, so it can't cross this cell membrane. So how does it exactly act on the target tissue? So it will act through the insulin receptors which are located on the cell membrane. So the insulin receptors, are having two alpha subunits and 2 beta subunits. So on the target cells there are insulin receptors, okay, that are the insulin sensors. So since this insulin can't cross the cell membrane, so it will act through the receptors which are located on the cell membrane of the cells, okay. So this insulin receptor has 2 subunits, that is the 2 alpha subunits and 2 beta subunits. So this alpha subunit is extracellular part, and this alpha subunit are connected by the disulfide bonds, and this alpha and beta subunits are also connected by the disulfide bonds, okay. So it has basically 4 subunits, 2 alpha and 2 beta. Basically it's a protein molecule, okay. It's a protein molecule with 4 subunits. So these 2 alpha subunits are located outside the cell membrane, that is the extracellular. So these are extracellular part of the insulin receptor. So this is a insulin receptor molecule, it is a protein in nature, okay. Now this beta subunit has some part that is outside the cell, it is extracellular, some part within the cell membrane, that is the transmembrane, and some part intracellular part, okay. So this is a insulin receptor molecule. Now this insulin from the blood vessel, it will bind, this insulin will bind to this receptor. This is insulin molecule, consider this as insulin molecule. It will bind to this insulin receptor and it will lead to some cascade of events within the cell, that is within some changes will be there in the cell wall, some changes will be there within the cytoplasm, and also some changes will be there in the nucleus. So insulin on its target cell, it will cause changes in the permeability of the cell wall, it's some changes in the cytosol, that is the metabolism machinery of the cell, and also on the nucleus. Okay, so how exactly this molecule without entering the cell, without entering inside the cell, just by binding the receptor outside the cell. So insulin molecule will never enter inside the target cells of the liver, skeletal muscle, and in particular adipose tissue. It will act on the almost all the cells, but in particular these are the target tissues. So it will never enter into the target cell, but it will bind to its receptor outside only, and it will lead to so many changes inside this cell that we are going to discuss under the mechanism of action. I hope till now this point is clear, that an insulin molecule will bind to the insulin receptors. Now, once the insulin molecule binds to the insulin receptor, that is the alpha subunits, which is extracellular, what happens is there is some transformational change takes place in the receptor. That change or signal from this alpha subunit goes to the beta subunit of the receptor. So in the beta subunit, there is one enzyme, that is intrinsic enzyme in the beta subunit itself. In the protein itself, there is one enzyme that is known as tyrosine kinase. Okay, so this tyrosine kinase enzyme, what it will do is, once the insulin is activated, that is insulin binds to the receptor, these enzymes which is present in the beta subunit gets activated, and once it gets activated, it causes phosphorylation of the tyrosine residues. That is, this is a protein molecule, so amino acids like tyrosines will be there, tyrosine amino acids. So the tyrosine amino acid of the beta subunit gets phosphorylated by this enzyme tyrosine kinase, that is phosphorylation takes place. Phosphorylation. Now this type of phosphorylation of the tyrosine residue of this beta subunit is known as autophosphorylation. That is, it itself, the intrinsic activity of this enzyme causes the phosphorylation of the tyrosine residue of the beta subunit of the insulin receptor. Okay, now this is autophosphorylation. Also this is a cross phosphorylation, that is the beta subunit of one side, tyrosine kinase activity enzyme of the beta subunit of the one side causes the phosphorylation of the tyrosine residues of the beta subunit of the another side. So it is cross autophosphorylation. Okay, so once this beta subunits get phosphorylated, then what happens is, inside the cytoplasm there is one protein. This is known as insulin receptor substrate, or IRS. Inside the cytoplasm at the resting condition it is present, but once the insulin binds to this receptor, insulin receptors alpha subunits, and there is autophosphorylation of this beta subunit, then this insulin receptor substrate protein now binds to this phosphorylated subunit. So the insulin receptor substrate, which is normally present in the cytosol, will now binds to this receptor. Okay, now what happens once it binds to the phosphorylated unit of the activated beta subunit of the insulin receptor? Now this insulin receptor substrate, okay, insulin receptor substrate or IRS, it itself get phosphorylated. That is, this is also a protein, and here also there are tyrosine molecules will be there in these proteins. So these tyrosine residues of this insulin receptor substrate or the insulin receptor molecule, they also get phosphorylated. Okay, again due to this intrinsic tyrosine kinase activity, now this also gets activated, that is the insulin receptor substrate, that is IRS. Now once it gets activated, it leads to some conformational change in this molecule, IRS molecule. So that conformational change in the IRS molecule will causes the formation of some active domain on this IRS molecule. Okay, when it is activated, so it gets activated, and there is some transformation, structural changes takes place after the phosphorylation of this IRS molecule. Okay, so once it is activated and there is some conformational change in this molecule, what happens is, now the main thing happens. So this is known as docking proteins. These are docking proteins. So it will dock the other proteins, or it will attaches the other proteins which are situated in the cytosol. So what happens is, on one side, that is in this docking protein, one protein or enzyme will bind now. Okay, on one side. So this enzyme which binds this, which is this insulin receptor substrate, it was free, but once it gets activated, it will bind to one more protein or one more enzyme. So this protein or enzyme, it is known as phosphatidyl inositol 3 kinase. Okay, it was in the resting condition of cytosol. Now it will bind to this one. Okay, so now this got bind, and this binding site of this phosphatidyl inositol 3 kinase, this is referred as SH2. This site, this binding domain of this, it is referred as SH2. That is known as Sark homology 2. Okay, but if you can remember, well and good, but concentrate on this molecule, this main enzyme, that is the phosphatidyl inositol 3 kinase. Now what does this enzyme, once it binds with this docking protein, this is called as anchoring protein, once it bind, it gets activated. Now once it gets activated, what it will do is, you know, on the surface, that is the cell membrane surface, this is lipid bilayer. So one of the lipid is PIP2, that is the phosphatidyl inositol diphosphate. Phosphatidyl inositol diphosphate. So this phosphatidyl inositol diphosphate, which is there in the, this is a normal lipid on the cell membrane, it gets converted into phosphatidyl inositol trisphosphate or triphosphate. So normally in the lipid cell layer, that is this one lipid will be there, phosphatidyl inositol diphosphate gets converted into phosphatidyl inositol triphosphate with the help of this enzyme, and this enzyme, it is known as phosphatidyl inositol 3 kinase. That is, it will phosphorylate bisphosphate into triphosphate, that is just present in the cell membrane. Now you can see this activities is just taking place within the cell membrane only. Insulin binded to the receptor, this receptor gets activated, it will bind to the insulin receptor substrate. There is a changes in insulin receptor substrate. This insulin receptor substrate bind to this phosphatidyl inositol 3 kinase, and this phosphatidyl inositol 3 kinase molecule converts this phosphatidyl inositol bisphosphate to trisphosphate. I hope till now the concept is clear. Now once this molecule phosphatidyl inositol 3 kinase 3 phosphatase is activated, or this molecule is formed, it has this phosphate residues. So to these phosphate residues, now two molecules get binded from the cytoplasm. Two molecules will bind to this molecules, okay, to this phosphatidyl inositol phosphate residues. Once it get converted, so now two molecules from the cytosol will get binded to this. Okay, one of this molecule, it is known as protein kinase 2, or simply AKT protein. Protein kinase 2 or AKT protein. Okay, one more is an enzyme. So this enzyme is phosphatidyl inositol triphosphate dependent kinase 1. phosphatidol inositol triphosphate dependent kinase. Usually kinase are the enzymes which will phosphorylate the other enzymes, kinases. So these are phosphatidol inositol triphosphate dependent kinase 1. So what this enzyme will do, it will phosphorylate this protein, that is the protein kinase 2 or AKT molecule. So it will phosphorylate or make it active. Okay, this was an inactive PK2 molecule, that is the protein kinase 2 molecule, protein kinase B molecule or AKT molecule. So it will get activated. So this is protein kinase B molecules. Protein kinase B, or also known as AKT molecule. So this protein kinase B gets activated or phosphorylated with the help of this enzyme phosphatidil inositol dependent kinase 1. Okay, so this gets activated. So once this gets activated, this PKB molecule, that is the main molecule or main protein, that is the protein kinase B or AKT molecule, which gets phosphorylated or activated. Now this molecule causes the changes within this cell, that is it will now help in reduction of the glucose from the blood, and how exactly it will take, now I'm going to discuss now. So before that, once again I would like to revise, since it's a complex pathway. So see, insulin is a larger protein molecule having a 51 amino acid, right? So this larger molecule and a polar molecule can't crosses this cell membrane from the blood. So this molecule with 51 amino acid, it will bind to the insulin receptors on this cell membrane. So once this insulin molecule binds to the insulin receptors on the cell membrane, which has alpha and beta subunit, and the beta subunit has a tyrosine kinase enzyme. So this tyrosine kinase enzyme causes the phosphorylation of the tyrosine residues in the beta subunit of the insulin receptor. So once this gets activated, insulin receptor substrate 1 proteins gets bind to this insulin receptor. After binding, this protein itself gets phosphorylated at the tyrosine residues. So once it gets phosphorylated, it leads to some conformational changes. So once this conformational changes takes place, then there is a binding. This is known as docking proteins. So some other proteins will come and bind to this insulin receptor substrate. So one of the important binding protein is phosphatidil inositol 3 kinase. Okay, so this phosphatidil inositol 3 kinase acts as an enzyme and converts the phosphatidil inositol bisphosphate or diphosphate into phosphatidil inositol triphosphate in the cell membrane. So once it is converted into phosphatidil inositol triphosphate, some two molecules will bind to this. So two molecules are phosphatidol inositol phosphatidil inositol triphosphate dependent kinase 1 and also protein kinase B or AKT protein. Okay, so this will get bind to this. So this molecule PDK1 causes phosphorylation or activation of this protein kinase B or AKT molecule, and this PKB molecule or AKT molecule will get removed from this or detach itself and comes into the cytoplasm and now causes the action. Now what action it will cause, I will tell you, but before that I would like to say this pathway, that is binding from the insulin up to this activation of this PKB molecule, this pathway, it is known as PKB pathway, that is phosphatidil inositol 3 kinase protein kinase B pathway. That is, ultimately when insulin binds to the receptor on the insulin receptors on the cell surface, outside the cell surface, it leads to activation of this molecule. Now this molecule is the main molecule which will cause us so many changes in these cells. What changes it will take to this cell? So in this cell, in the cytoplasm, you know, in the cytoplasm there are vesicles. in the target cells, especially these striated muscles, that is skeletal muscles and cardiac muscles, and also in the adipose tissues there are vesicles, and in these vesicles there are certain glucose transporters, certain channels, ion channels, glucose transporters. So this type of glucose transporters which are present in the cytoplasm of the skeletal muscles and as well as adipose tissue, these glucose transporters are known as GLUT4, glucose transporter 4, and this is insulin dependent glucose transporter. So this activated protein kinase B, or the phosphorylated protein kinase B, or ACT protein, what it will do is it will stimulate this vesicles, it will push this vesicles. First action of this protein is it will drag this vesicles towards this cell membrane, and this cell membrane now will have this GLUT4 receptors, GLUT transporters. So because which was there in the form of vesicles inside the cell, now they are dragged to this towards the cell membrane, and once it binds to the cell membrane, this will causes the entry of the glucose molecule from the blood. So generally glucose molecule is also a polar molecule, can't enter directly, so it has to be transported through the glucose. So the first action is glucose uptake in the skeletal muscle and adipose tissue. In liver there are GLUT2 receptor, that is insulin independent. We'll discuss in the actions. Right now just remember these GLUT4 molecules in the skeletal muscle and adipose tissue. So the first action of this insulin is it will cause glucose uptake, that is from the blood glucose enters because it will increases the GLUT receptors on the cell surface, and glucose molecule will enter inside this cell. This is one action. First action, that is the glucose uptake. Second, once the glucose gets enter into the cytoplasm, so this glucose molecule gets converted into glucose 6-phosphate by the enzyme known as glucokinase, in the liver cell, and also known as hexokinase in the skeletal muscle and adipose tissue. So this molecule will stimulate this glucokinase enzyme and gets converted glucose into glucose 6-phosphate. So this glucose 6-phosphate now gets converted into glucose 6-phosphate, and ultimately pyruvate is formed, and from that ATP is released, that is glucose utilization. So first action of this protein kinase B or AKT molecule is glucose uptake. Second action is it will convert glucose to ATP, that is the it will cause us increased utilization of the glucose inside this cell. Now also increased utilization, now this will convert glucose into glycogen. How it stimulate the glycogen synthase we'll discuss in the next video, that is the actions of insulin or carbohydrate, protein and fat metabolism. Timing, remember how it will do. So the mainly it will do through this activated protein kinase B or ACT protein. This is the one pathway. So what it will do is, here this pathway, it will increases the permeability of the cell membrane, that is it will affect the cell membrane, also the metabolism in the cytosol, that is it will increases the glucose uptake, also it will convert the glucose into glycogen. Also it will convert the glucose, or it will take also the amino acids. When it is taken, it will lead to the protein synthesis and lipid synthesis. That are the different actions we will discuss. So main in the mechanism, just remember the most important thing is this one, this pathway PI3, phosphatidol inositol phosphatidol inositol 3 kinase, and PKB, that is protein kinase B pathway. This is one action. So with the help of this, it will alter, insulin will alter without entering the cell, it will enter the cell membrane and cytosolic enzymes by just simply phosphorylating various proteins. Now apart from this metabolic action, insulin also has this action on the growth of the cell, that is cell proliferation and mitosis and cell differentiation. So it will act on the nucleus also. So apart from this general metabolism in the cytoplasm, insulin molecule has an effect on the nucleus, for it will causes the mitosis, cell differentiation and cell proliferation. So how exactly now this insulin molecule will act on the nucleus? So it will act on the nucleus by another pathway, so that pathway we'll discuss now how exactly it will go there. So again, this pathway is similar, that the next pathway which will act through the nucleus is also similar. That is, once the insulin binds to the insulin receptor, so alpha subunits, there is some transformational change which will activate the beta subunit. Inside the beta subunit there is a tyrosine kinase activity, it will phosphorylate the beta subunit of the receptors of the beta subunit of the insulin receptor, which will again bind to the insulin receptor substrate. Okay, so on another side of this insulin receptor substrate, one more protein will bind. Okay, so this protein is known as GRB2. Okay, GRB2. Now it will bind to this protein. Okay, growth factor binding protein. Okay, GRB2. Now after when it binds, so this again it is binded because this protein also has this similar structure or this domain, that is the SH2 domain, that is the Sark homology 2 domain, which will bind to the insulin receptor substrate. So once it is binded to this IRS substrate, what will happen? Again, a series of reaction takes place. So that series of reaction will lead to activation of the RAS protein and MAP kinase. So this pathway which will activate the insulin, which will activate the receptor and activate the nucleus. So that is known as RAS MAP kinase pathway. So it will now bind this GRB2 protein. Once it gets binded, it will get activated. So this activated protein now will again activate one more protein molecule known as SOS. So this, the name was given as son of 7 less. Okay, don't concentrate much on the names, just remember one protein, GRB2, and then it will here the Sark homology 3 domain will be there. So this GRB2 protein will activate this SO2 molecule, and this SO2 molecule, SOS molecule or protein molecule now will activate this RAS protein. Okay, so this RAS protein, which is binded to the cell membrane. So this RAS protein is usually at the resting condition, it is inactive state because it is binded with one molecule, guanosine diphosphate, GDP. It is an inactive state. So once this sequence of events takes place and this SOS molecule is activated, this SOS molecule causes the activation of this RAS molecule, and this RAS molecule now gets activated. So this RAS molecule will now, instead of GDP, the activated molecule will have guanosine triphosphate. That is, this activated SOS molecule will causes the activation of this RAS protein. Okay, so this activated RAS protein will have GTP, guanosine triphosphate. Okay, now this activated guanosine triphosphate will lead to some series of RAS molecule, activated RAS molecule, that is which contains GTP, will lead to some series of reactions. So many proteins, intermediate enzymes and all will lead to take place. Ultimately there is activation of mitogen activating protein kinase, MAP kinase. So this MAP kinase will enter into the nucleus and it will activate the MAP protein, that is the mitogen activating protein. So it's a group family of proteins, okay, basically growth factors. So it will activate this MAP, and this activated MAP will now stimulate the genes, that is DNA. Then there is a formation of the messenger RNA which will come into the cytoplasm and will leads to the translation process and the protein synthesis takes place. Okay, this is how the insulin will act on this nucleus. Okay, now one more point I would like to bring here is, this is general growth factor pathway. This is general growth factor pathway. So in other cells where this growth takes place, the pathway is similar. Instead, there will be no, this insulin receptor substrate will not be there. So what happens in neoplasia or cancer? There is increased activity of this RAS GTP molecule, that is activated RAS molecule, and they will continuously stimulate this nucleus and continuously cell proliferation takes place. So there should be one break that should stop this activated RAS molecule once its action is over. So that breaking molecule, okay, so that breaking molecule, it is known as GAP. That is GTPase activating protein. So that will inhibit this molecule and it will stop it, so that whatever desired protein is released, the message is released, it should synthesize only that proteins only. Okay, it should not continuously synthesize the proteins. If it is continuously synthesized, that is one of the cause of the cancer or the neoplasia. So this GAP protein will inhibit this activated RAS molecule. Okay, so this is how mechanism of insulin takes place, basically by the two methods, PI3K, that is the phosphatidylinositol 3 kinase and protein kinase B pathway, and RAS MAP kinase pathway. So this RAS MAP kinase pathway will stimulate the nucleus, and whereas this pathway will stimulate the cell membrane and cellular organelles. So here this mechanism of action, the action, that is the cellular event, that is from this GLUT4, the glucose molecules will enter into the target cell within seconds. So time duration is within seconds to minutes. Within seconds to minutes, it will, this insulin molecule show its effect within seconds to minutes on the cell membrane. Then from minutes to hours in the cytoplasm, it will take minutes to hours in the cytoplasm for all that glycogen synthesis, glycolysis process should take place within minutes to hours, and finally this, it will take hours to days. So this pathway will take hours to days. Okay, so this is the mechanism of action of insulin release. So stay tuned with us and please subscribe our channel for our next video on actions of insulin and diabetes mellitus. Thank you very much.